In this video, we're going to look at reactions that are related to the citric acid cycle. And the citric acid cycle is what we call amphibolic. That is, it has both anabolic and catabolic reactions that are related to the cycle. And we're going to start with the catalytic reactions, that is, reactions that use and drain our citric acid cycle intermediates. One we already talked about is gluconeogenesis or glucose biosynthesis. We can take oxaloacetate and we convert it to malate or aspartate. Once we convert oxaloacetate, it can be moved out of the mitochondria and can be used in gluconeogenesis. And we already talked about this in a previous video. We can also use intermediates to make fatty acids. Here we need acetyl-CoA, but there's a problem in that acetyl-CoA cannot be transported out of the mitochondria. Therefore, acetyl-CoA is converted to citrate, and then citrate can be converted outside the mitochondria to make our fatty acids and our cholesterol. Lastly, we can make amino acids, and we have either alpha-ketoglutarate or oxaloacetate that makes this for us. And here's just an example of an alpha-ketoglutarate reaction that makes us an amino acid. So alpha-ketoglutarate plus NADH plus NH4 will make glutamate for us. Now let's look at the anaplerotic reactions that will replenish our intermediates. First, let's look at the enzyme pyruvate carboxylase. We can take pyruvate and add a CO2 to it with ATP and water to make oxaloacetate. And this is the first step of gluconeogenesis, that is taking pyruvate and making it into oxaloacetate. This enzyme is actually activated by acetyl-CoA, which might seem a little weird because these reactions are making intermediates. So you might be thinking, if we have a lot of acetyl-CoA, shouldn't we have a lot of intermediates already? Well, what happens is if we have a lot of acetyl-CoA around, that means we're lacking in intermediates. We can't run the cycle efficiently. So we have a buildup of acetyl-CoA. So that acetyl-CoA is going to go and activate pyruvate carboxylase, and then it will make oxaloacetate. Then oxaloacetate and acetyl-CoA will combine, and this cycle will run. And this increase of our intermediates will actually speed up the flux of how fast our acetyl groups can go into the cycle. By just taking pyruvate and making it into like an oxaloacetate or an alpha-ketoglutarate, we can speed up the overall flux. For example, 60 to 100 times faster in muscles during exercise if we divert some of the pyruvate away to make an intermediate instead. As I alluded to, pyruvate can also take an amino group from glutamate, and that pyruvate will be turned into alanine. Glutamate will be turned into alpha-ketoglutarate. Then again, we have intermediates. And all of these reactions are going to speed up the citric acid cycle, which will increase the amount of ATP that we produce. Now, one other cycle we need to talk about, the glyoxylate cycle. And that's because it shares some steps with our citric acid cycle. So plants, bacteria, and fungi have special enzymes that allows them to convert acetyl-CoA to oxaloacetate, which is one of the steps that you use to make glucose, gluconeogenesis. In plants, we call these enzymes being a part of the glyoxylate cycle. And this operates both in the mitochondria and the glyoxysomes, which are this just specialized plant peroxisomes. And there's 5 reactions for the glyoxylate cycle. So reaction 1 and 2, glyoxysomal oxaloacetate, so oxaloacetate in the glyoxysome, is condensed with acetyl-CoA to make citrate. And then a conatase can take this citrate and make it into isocitrate. So that's the first 2 reactions. Then we can take this isocitrate and using glyoxysomal isocitrate lyase, this will be cut to make succinate and glyoxylate. Now I'm going to take a little detour here. This succinate can be transported to the mitochondria and go through the citric acid cycle to make malate. This malate can be transported into the cytoplasm for oxaloacetate and gluconeogenesis. So that's where one of your products is going. Your other product, glyoxylate, will continue through the glyoxylate cycle. So reaction 4, malate synthase, will condense glyoxylate with acetyl-CoA to form malate. And then reaction 5 is malate dehydrogenase, taking malate and NAD plus to make oxaloacetate and NADH. And so this oxaloacetate can go through the cycle again to make another malate oxaloacetate into gluconeogenesis. Or this oxaloacetate could be used for other things as well. Overall, you use 2 acetyl-CoAs, 2 NAD pluses, and FAD. And what you make is 1 molecule of oxaloacetate, 2 coenzyme A's, 2 NADH, plus FADH2 and 2 hydrogens. So basically, instead of making carbon dioxide, you're going to just make additional oxaloacetate, which can be used to make glucose. Fortunately, humans don't have this pathway. This is just seen in plants. And with that, that ends that video, and I will see you in the next one.